Hello everyone. In this video, we discuss the basic introduction of chromatography, its application, and the basic principle used in chromatography. First of all, what is chromatography? Chromatography is a technique for separating mixture into their components in order to analyze, identify, purify, and quantify the mixture or component of the mixture. Here in diagram you can see the mixture is shown, and later after chromatography the mixture is separated to its component. Uh, let's come toward the uses of chromatography where we can use chromatography. Chromatography is used scientists for several purposes, in which some purposes are discussed here. The first purpose of chromatography is to analyze, examine a mixture, its component, and their relation to one another. uh, Identification, or we can say identify, is the second purpose of scientists. uh, Chromatography is used to determine the identity of a mixture or component based on known components. Next one is purify, separate component in order to isolate one of interest for further study. Next one is quantify, determine the amount of the mixture and are the component present in the sample. Now, the application of chromatography. Real life example of uses of chromatography. The first one is pharmaceutical company. In pharmaceutical company, we use chromatography to determine the amount of each chemical found in new product. In hospital, we use chromatography to detect blood or alcohol level in the patient bloodstream. Uh, in law enforcement, we use chromatography to compare a sample found at a crime scene to the sample from suspect. Environmental agency, we use chromatography to determine the level of pollutants in the water supply. In manufacturing plant, we use chromatography to purify a chemical needed to make a product. Definition of chromatography. Basically, chromatography is a laboratory technique that is separate component within a mixture by using differential affinities of the component of the mobile medium in for a stationary absorbing medium through which they pass. The terminologies used in chromatography are differential. Differential is basically showing a difference distinctive. Affinity is a natural attraction or force between things. Mobile medium, uh, it may be gas or liquid that carries the component of mobile phase. Stationary medium is the part of the apparatus that does not move with the sample. A simplified definition of chromatography is chromatography separate the component of a mixture by their distinctive attraction to the mobile phase and the stationary phase. If we look at the explanation, a compound is placed on stationary phase, which is used to be separated. Mobile phase passes through the stationary phase. Mobile phase solubilize the component, which is usually a solvent. Mobile phase carries the individual component a certain distance through the stationary phase depending on the attraction of both the phases. Uh, Here is the example in which a mixture is shown and later this mixture is separated into its component. The yellow one is separated, the red one, and also black and blue one. Here is a detailed diagram of chromatography. Now, there are certain types of chromatography. Liquid chromatography. Liquid chromatography is usually separate liquid sample within with a liquid solvent mobile phase and a column composed of solid bead stationary phase. In gas chromatography, separate vaporized sample with a carrier gas mobile phase and column composed of a liquid or of solid bed stationary phase. Vapor chromatography separate dried liquid sample with a liquid solvent mobile phase and paper strip stationary phase. Thin layer chromatography is separate dried liquid sample within a liquid solvent mobile phase and glass plate covered with a thin layer of alumina or silica gel stationary phase. Principle of paper chromatography. Uh, there are two principles of paper chromatography. The first one is capillary action. The movement of liquid within the space of a porous material due to the forces of adhesion, cohesion, and surface tension. The liquid is able to move up the filter paper because its attraction to itself is stronger than force of gravity. Solubility, the degree to which a material solute dissolve into a solvent. Solutes dissolve into solvent that have similar properties, like dissolve-like principle. This allows different solutes to be separated by a different combination of solvent. Separation of components depend on both their solubility in the mobile phase and their differential affinity to the mobile phase and stationary phase. In next video, we will discuss the experiment of paper chromatography. Thank you for watching.